Hello all, it's Dr. B, and here we go for the last time. This is major parts of Chapter 11 that would require just a little foundational understanding, so let's get to it. The first example of what I want to talk about a little bit is something called QFPCR. It basically stands for quantitative fluorescent PCR. And what you're really doing is you are amplifying a known genetic marker or an STR, which is a small tandem repeat, on chromosome specific DNA. And so the difference here is that primers are labeled with a fluorescent marker and amplified to determine copy number. And we use this mostly for trisomy. So if you think that you might have, a child of yours might have Turners or Klinefelters, or it's way better on non-X and Y chromosomes. But for example, trisomy 21 is a good example. Down syndrome. So like how does it work? So here's how it works. So basically, this would be a picture of kind of what the outcomes would be, the different versions of what you'd see in terms of the results. So genetic markers or STRs, they may vary in length between individual chromosomes and people, depending on the number of repeated STRs that you have. And so the relative copy number is determined by calculating the ratio of the peak area or the peak height for each marker. So if we take a look at like this first one here, this would be a normal heterozygote. You'd have one copy of each allele. And you can see here that you have two different peaks. Each of these would be a different fluorescent dye. Hence, you have definitely a heterozygote. So this would definitely be a negative for a trisomy. And so remember that a normal diploid sample has a contribution of two of each of the investigated chromosomes. And so two alleles of a chromosome-specific marker detected as two peaks is a 1-to-1 ratio. So this is a case, again, when the marker is heterozygous, and that's why you see what you see. So if we take a look at this second one here, we have one peak that's twice as high as the second peak. And that's because this first allele, there's two copies of it. Thus, it fluoresces at twice the fluorescence that one copy would fluoresce. And you even have the second one as a relative reference height. So because this one's twice as high as this one, you have a 2-to-1 ratio, which indicates a presence of an additional marker copy that might correspond to an additional chromosome. And so we see another version here, which would be trisomy as well. And we can see because we have three allele peaks here. So this would be somebody who would have three copies of the same allele, where this would be somebody who would have two copies of one allele and one copy of the other allele. What I would not do in an exam is ask you about this one. And the reason why is the marker analysis would not be able to distinguish between a homozygous or a monosomic situation. So that's generally considered not informative. But I would definitely ask you about the three that you would see here. And so am I just going to be giving you pictures of heights? The answer is no. I'm going to give you something like this. And this is the peak areas that would pertain to a picture like that. And you can see here that this is one way I could ask you. So actually, yeah, go back on that one point. I could just basically circle something like this and say, would this be a trisomy or not? And the answer is absolutely, because you can see this height is twice as high as this height. So there's two copies here, one copy here. And that's definitely a trisomy. These are a 1-to-1 ratio of height, 1-to-1 ratio of height, 1-to-1 ratio of height, 2-to-1 ratio right there. And then we have one that's a 1-to-1-to-1 ratio. So these three would be considered trisomy. The way that I would in all likelihood do it, just because it's easier on exam, would be to give you a chart kind of like this, which is in your book. I won't give you this exact one. But really what you're looking at is the relative difference in these numbers. You can see here 16,705 is really close to 14,667. They're almost a 1-to-1 ratio. And so obviously this is one I would not ask you, because you can see it's just one peak. So if we take a look at, for example, if we go back to this chart and take a look at, let's see, D21S11, we see D21S11, we see here that there's a 2-to-1 ratio, and the reason why you can see is that 28,000 is twice as much as 12,000. So there's a double peak here and a single peak here, and you would be expected to know that that would be trisomy. The other example would be the D21S1411, which is this one, much easier to see because obviously there's 3 peaks, and they're all at a 1-to-1 ratio. So that is how you would determine on an exam the difference between the peak heights and what they mean. You'll have a few questions on that. Spend a little time getting into some of the testing techniques that we can use for some of these things. So this that you're looking here is basically called FISH, and it's referred to as fluorescence in situ hybridization. And basically what it is you have a labeled DNA clone of interest is hybridized to a chromosome and treated with a DNA denaturing agent. And so when we do this in a metaphase state, we can see a signal representing the hybridization of the target sequences on some of the sister chromatids. So we can use this to detect, for example, like in this case, this is looks like a deletion, because you'll notice that there's a color missing on one of the chromosomes. So the general idea here again is we have a clone, we label, and if indeed the complement is found on the chromosome, it will fluoresce. So we can make these signals kind of whatever we want based on whatever sequence we have. Another test is called Southern blot hybridization assays, and you're probably familiar with Western blotting, how we can identify a particular protein using antibodies. This one is Southern blotting, and so again there's also Northern blotting as well, but we can use this to detect mutant alleles, and basically what you see here is we have DNA that's transferred to a membrane, and then we use a hybridized probe. So basically we would use the opposite complement of what we suspect we're looking for, and that probe will stick to its complement from the sample, and then we can use either X-ray film or whatever other kind of chromogenic way to see whether or not our probe is there. And if our probe is there, it means it's stuck to a complement that would reflect what it is we're looking for. So it's one of the ways that we can identify. You can see here, this would be an example of a probe. The markers lanes are here, and we can see that there is the presence of whatever we were looking for in the sample of DNA. This is a quick little intro to NGS, which is next generation sequencing, and this is definitely where most of the work is going because it's becoming cheaper and faster. But it's a way that we can analyze large quantities of DNA when we don't exactly know what we're looking for, in the sense of you know, we might be looking for one thing, but there may be other issues somewhere else, and unless we're looking specifically with a pair of primers, we're not going to find them. NGS is different. It basically gives you readout of the entire DNA sequence, and I have an Edpuzzle on that, so you definitely want to watch that as well. So basically you take genomic DNA and you chop it up and you add little adapters to the end, and then these things are stuck to a certain bead, and then those beads are stuck to a particular slide, and then each of the little pieces are sequenced. And then what happens is, once these little pieces are sequenced, the sequencing happens by the cleavage of a fluorophore, which is a chemical and each letter is given a different fluorophore, A, T, C, and G. And when this is done over and over again, you get lots of little teeny reads, like down here. But then a computer will overlap all of these reads into one gigantic long, millions long read. And then that information can be used with bioinformatics to, using NCBI and other databases to just basically say, hey, let's find this gene. What's going on here? Do I have the proper sequence, or what sequence do I have? So you definitely want to check the Edpuzzle out. But NGS is absolutely going to be the future of a lot of the PCR-based replicative technologies we're going to use to identify genomic information. A more fundamental type of sequencing is called Sanger sequencing. I actually do this a lot in my own work. It's also become way cheaper. This can be used to sequence generally DNA strands like around maybe 70 to 80 on the lowest end of base pairs, up to maybe 1,000, maybe 1,500 base pairs on the high end. So this would be where you've already kind of have isolated a particular gene or area of interest, and you just want to sequence that. We use this a lot for identification of organisms, and a lot of things in medicine, especially microbiology and healthcare, are moving towards this. Many people think that the days of MLTs, just plating organisms from a patient sample on different types of selective and differential media, and doing all kinds of TSI or MSA plates or chocolate agar. Those days are quickly ending, because all of those tests exist to identify a particular organism. We now have this technology which is cheap enough and fast enough, and I wouldn't be surprised within the next 10 years, while some of you are MLTs, that a lot of your testing that's done for bacterial identification goes to a MALDI-TOF instrument or another type of genetic analyzer. So how would this basically work? You basically do a regular PCR at first with primer, but you really only use a forward primer. The difference is that you have a few radioactive dideoxynucleotides. So you have your regular A's, T's, C's, G's in there. But you also have some that are radioactively labeled and also will cause chain termination. And so if you have PCR happening over 40 or so cycles, randomly the polymerases are going to be pulling from the environment and incorporating at random places these fluorescent nucleotides that will stop termination. There's two different ways that you could analyze this. The old school way is through dideoxy sequencing on a gel, where you would basically do the same PCR in 4 different tubes, but you'd only put fluorescent A's in one tube and C's in another and G's in the third and T in the fourth, with the normal A's, T's, and C's and G's. Again, this first tube would have normal A's, T's, C's, G's, but also fluorescent A's. And basically, from that point, you would then run a gel and read this from the bottom up, because obviously the shorter base pairs move faster in a gel. So this particular sequence would be read as G, A, C, T, G, A, G, C, T. Now, the newer instruments don't do that. They do it automatically. They have a capillary. That's the instrument that I have, and as soon as I'm done the PCR, I run it into this capillary instrument, which sucks up all the fragments and lines them up in order, and a laser reads these. So if you see that this is a race, you can clearly see that this first G, one of the polymerases, happened to incorporate a G at the very first nucleotide, because that was the first nucleotide, was the C, the opposite. And so the machine lines them up and just reads the laser in order. You can see here GACT, bam, right down the line. So that's generally how that works. It's very efficient. It's very fast. If I were to set up a PCR at like 10 in the morning, I would actually have the sequence, the full reading of the genome sequence of even, say, who cares, maybe a 600-700 base pair fragment. I would have that information by about 3 in the afternoon, which is way faster, and it's getting cheaper as well. So once we get all this information, it's just one example of a particular um, bioinformatic tool that we could use to analyze the results. We can basically take whatever are either in this case it would have been DNA, um, and if we use something like a cDNA, which again is going to be only the particular nucleotides involved in protein synthesis. So even though originally what we sequenced was the DNA, we can figure out exactly what the amino acid would be as well if we use, for example, like a cDNA from mRNA. So we can see here that a normal, in this particular case right here, there should be a cysteine at this particular location. But we find out through the sequencing that there's actually a tyrosine right here. And so one little side thing that I can add for how you might write that, for example, would be, if I just bring this in here, how would we indicate that particular scenario? First thing we do would be that. Any idea what that might stand for? Well, that stands for the fact that we're looking at protein. The next thing we do is we write what the normal version would be, which would be cysteine. And then the location is the 231st amino acid, and then what actually it became. So we can read that this is a mutation in the protein of cysteine at amino acid 231 into tyrosine. And so that's just another little trick for you. Another test is one that is called the ARMS test, and basically it stands for Amplification Refractory Mutation System. And basically what that means would be this. So let's just say this is your DNA, and this is both strands of the DNA, and you're doing a PCR. And obviously during your denaturing stage, here is the one strand of DNA, here's the other, and you are going to have then your primer annealing, which is going on here for both of these strands. And you can see that this particular primer right here has a G at the very end of it, the 3' end of the primer. Well, that is absolutely complementary to the normal wild type case of what someone should have. So you will see what's going to happen here. Obviously, since there is a normal wild type C in the last position and there's a G there, amplification will happen. Now, and again, this is done by designing primers that end right where that SNP would be, single nucleotide polymorphism. So now let's take a look at, and again, we don't know what this person is yet, whether it's the green or the red. And so now let's just repeat the same process. Now here's the DNA. We separate them through the denaturing step, and now we have the primers annealing, but you'll notice that on this strand, this is right where the single nucleotide difference is. And so because of that, the T's disaffinity for the G, DNA synthesis isn't going to happen, and you are not going to get amplification. So if there tends to be a germline or a noted substitution that we just know of, for example, like sickle cell is always going to have the same substitution. The third step would be done on all these, which is to create a reverse primer. There's your forward primer, there's your reverse, whatever. They really don't mean anything. They're just ways to tell them apart. They will also do another PCR with the incorrect mutant letter at the very end, which would be the A, and therefore it would cause amplification and further evidence that that person has that particular singular nucleotide difference. So it's a pretty cool test. I've actually done it a few times, not with people, just done the test, just to see how it works. It's really cool. I've done it with other organisms. So that is just a recap of major aspects of Chapter 11. And again, if you have any questions, feel free to contact me. All right, bye.